In step 4, we're going to discuss causing damage or disease in the host. There are 3 ways microorganisms can cause damage to the host. Enzyme action, which is a form of direct damage. Toxin action, which again is a form of direct damage. And then the third one is indirect damage that's caused by the host's defenses being excessive or basically reacting inappropriately. First thing I mentioned was enzyme action. Many microbes produce extracellular enzymes that are also known as exoenzymes that can damage host tissues. These enzymes are typically linked with the spread of the bacteria to other tissues. There are 4 common microbial exoenzymes. This is the example I have. Here's a bacteria releasing its enzymes, and it's damaging the tissue enough that it's creating an entrance for this bacteria into the body. We have 4 that we're going to talk about: mucinase, hyaluronidase, coagulase, and kinase. Mucus is something that lines a lot of the cavities that are exposed to the external environment. Our digestive system is lined with mucus. Food goes from out here to in here, so digestive system is lined with mucus. It's a way to coat the cells and hopefully catch things without letting them get past. If you are bacteria and you have mucinase, you can dissolve that mucus, which leads to not having that nice protective coating anymore because it dissolved away. Hyaluronidase digests hyaluronic acid. This is the cement that holds our cells together. This is, an enzyme of, this is an example of probably hyaluronidase. Hyaluronidase takes the things holding our cells together and breaks them down. That gives me an entrance as a bacteria. Coagulase and kinase kind of do opposite things. Coagulase causes the blood and blood products to coagulate, forming a wall between the immune system and the bacteria, giving the bacteria a safe space to keep multiplying and doing what it's doing. Whereas kinase, the one down here, is going to dissolve clots instead of making them. Why? A lot of times when there is damage to our body, we will clot the end of, let's say, an artery or vein or whatever, a capillary. The reason we do that is because our body is making a wall so that the bacteria can't get past the wall. If I can dissolve the wall, that again gives me entrance into the body. Toxin action. Toxins are specific microbial protein products that are chemical in nature and may be poisonous to other organisms. Toxins are named according to their target. So if I have something that attacks the brain, the spinal cord, the neurons, I call it a neurotoxin because it's going after nervous tissue. We have 2 major types of toxins: exotoxins and endotoxins. Exotoxins are produced by microbes and secreted from the cell, and they have a strong specificity for a target in the host, a specific target host cell. I am zeroing in and getting to that place, that tissue. It can be very powerful. It may be lethal depending on what it is. It primarily works by damaging the cell membrane and disrupting the cell's function. In the example, we have Clostridium tetani secreting an exotoxin, tetanospasmin, that actually has the target of our spinal cord. It's being secreted to go to a specific place to attack a specific target. It's not general, it knows exactly where it's going. Even though in the example, this came in through the foot, it doesn't matter, it's going to the spinal cord. Endotoxin is different, and it involves a component in the cell walls of gram-negative bacteria, the LPS. We talked about that when we talked about the cell walls. It's released kind of unintentionally when our immune system breaks down these cells, when it actually kills them. Parts of the cell wall, as it breaks up, cause systemic effects in your body. Your immune system reacts to these toxins being present in your body. It can cause fever. That's systemic. You don't get a fever, in your pinky toe. You get a fever systemically all over your body. It can cause inflammation. And if that happens all over your body, you're going to be in a big, big bind, because it can close off your breathing tubes. It can close off, it can inflame your lungs. It's a bad idea. It can lead to sepsis, which is a blood infection, or ultimately, if your immune system gets way too carried away, it can actually lead to death. It can throw you into shock and lead to death. This is the endotoxins. Let's compare them, exotoxin to endotoxin. As far as toxicity goes, exotoxins can be toxic in very small amounts. Endotoxins, it kind of has to be a high dosage. What does it affect? Like I said, exotoxins have a specific target. Endotoxins, it's body-wide. You've got systemic effects like a fever or inflammation. Exotoxins, chemical composition, they're small proteins. Endotoxins, they're lipopolysaccharides that are found in the cell wall. Remember that LPS we talked about? They are specific to that gram-negative cell wall, something that our body doesn't normally see. It's not something that's in our body. Exotoxins, can they be denatured by heat? Yes, they can. Endotoxins can't, which is a bad thing. Toxoid formation. Exotoxins can be converted to a toxoid. Endotoxins can't. The immune response, as far as exotoxins go, it's antitoxins. It stimulates the formation of antitoxins. Endotoxins don't. Exotoxins don't normally cause a fever. Endotoxins do. Exotoxins are released from the cell itself. They're making a product and releasing that product. Endotoxins, when the cell gets torn apart, it gets released because it's part of the cell itself. Typical sources, the exotoxins, there are a few gram-positive and gram-negative that release these. With endotoxins, anything that's gram-negative that gets ripped apart by lysis, it's going to release the lipopolysaccharide from the cell wall. When the damage is indirect, caused by your own immune system. This was the third way that we said could cause damage. Many diseases are literally the result of indirect damage to the host. Indirect damage is a consequence of the interaction between the microbe and the host. The relationship between the two can sometimes cause the host's immune system to completely overreact, or to even react inappropriately, which can lead to damage all by itself. In the example here, we have an encapsulated Staphylococcus pneumoniae. Remember what I said about the capsule? It can protect it, prevent the immune system from working. With that in mind, your immune system keeps getting the signal. There's bacterial presence here. There's bacterial presence here. And it may have sent something to deal with it, but since it didn't work, it gets another signal, so it sends more, and it sends more, and it sends more. As this continues, you're going to get things like inflammation. You're going to have fluid release in the lungs. And if you've got fluid filling your alveoli, you're not going to be able to breathe. The fact that you have something that is encapsulated and something that remains there for a long period of time means that your immune system will keep reacting to it, keep reacting to it, keep reacting to it. It's not something that it kind of goes, oh no, it's not working, so I need to do something. It just keeps reacting to it. The process of inflammation involves fluid edema. Basically, it swells up and fills with fluid. That's happening in your lungs. You're going to suffocate. You're going to drown. This can be dangerous. Now, is it the bacteria's fault? Indirectly, yes, but it's your immune system that's doing the wrong thing. We have several different types of infections. Localized infections are exactly what they sound like. Microbes enter the body in a specific place and they stay in that area. They don't go anywhere else. Things like pimples, boils, warts. That's what that is. A systemic infection means that you've got microbes that come in maybe in one spot, but then they spread to all of your systems. That's why it's systemic. They spread to several bodily sites. Usually they'll do this via the bloodstream. Chickenpox works this way. HIV works this way. I know everybody goes, well, chickenpox only stays, on your skin, you get the sores. No, actually, it ends up staying with you forever, and it goes into a series of neuronal ganglia around your spinal cord, and it can reactivate into shingles as you get older. A focal infection. Microbes or the products of them spread from a local site and they're carried to other specific tissues. Remember, I talked about tetanus. You may, get a nail in your foot, but the actual titanospasmin, that exotoxin, is going after your spinal cord. So I may come in one spot, but what I'm going to affect is in another spot. Sorry. A mixed infection, also known as a polymicrobial infection. Poly means many. So ultimately, when you see polymicrobial or mixed, it means there's more than one bacteria there, or more than one invader. Several species of microbes establish themselves simultaneously at the site of infection. It's not just one thing, it's a bunch of things. One of the worst offenders is a human bite. We have a lot of things in here, not just one. So when we bite somebody, we are infecting them with a lot of different things. Wound infections. Depending on what you got wounded with, it could be something potentially that has multiple things on it. Other terms, a primary infection, a secondary infection. Primary is the initial infection. Primary means one, so it's the first thing that's there. Any type of infection that sets in first. Secondary, just like number 2, secondary. It's a second infection caused by a different microbe than the one that it was involved in the primary infection. It is often the result of the lowered immune defenses of the host. I'm already sick and I get exposed to something else, it's going to take advantage. So if I have the flu, Streptococcus pneumoniae may take advantage of that and also infect me. An acute versus a chronic infection. An acute infection, he's so cute. No, I'm just kidding. An acute infection basically means it's short. Okay? You've got rapid onset of the infection, severe symptoms, but normally they're short-lived. They don't last for very long as far as not only your symptoms, but the effects. The flu is an example of this. You get the flu, it lasts for 7 to 14 days, which is relatively short. It's not years. But after that 7 to 14 days, you're over it. Short. Chronic infections are different. They infect and progress and persist. They don't go away. HIV is an example of this. If you get HIV, you will have it forever. It does not go away. So other terminology associated with an infection: a sign, a symptom, a syndrome, asymptomatic, subclinical, inapparent. A sign. This is objective evidence of a disease noted by an observer. If I have a skin infection, you will see a boil, or you will see a pimple, or you will see something observable on that patient that there is something happening. It isn't, I feel bad. I literally, as a doctor, can observe what's there. That's a sign. Symptom. Subjective evidence of a disease sensed by the patient. If you tell me my stomach hurts, I can't prove that as a doctor. I can't go, oh yes, I feel that. If you tell me my ankle hurts, I'm not sure why. I can hear what you're saying and you're presenting me with a symptom, but I can't see that. So a sign is something I can see as a doctor. A symptom is something you're telling me, but I can't see it, so I can't prove it exists. Syndrome. It's a disease identified by certain complex signs and symptoms. If somebody has multiple sclerosis, there are a whole series of things that a doctor checks for to narrow the diagnosis to multiple sclerosis. There are specific signs that they will see, and there are specific symptoms that the patient will mention that will kind of drive to that. Is it a perfect science? No, most of the time it's not. Multiple sclerosis gets misdiagnosed all the time, and it could either be named as another disease, or it could be another disease named as multiple sclerosis, because a lot of the signs and symptoms are similar to each other. Asymptomatic, subclinical, or inapparent. An infection that lacks noticeable symptoms. You have an infection. There are bacteria or virus, whatever, in your body, but you don't show it at all. You don't feel bad. People can't see it. A good example of this is Typhoid Mary. She had typhus. She was a nurse. She knew she had typhus and was capable of spreading it, but outwardly there were no signs, there were no symptoms. So she kept working in hospitals and infecting her patients. And as people kind of started noticing that typhus was spreading around a hospital, she would quit and go to another hospital where they didn't know her. Terminology associated with inflammation. Edema. Edema is a fancy way of saying swollen or swelling. So what is happening in edema? Edema is actually caused by fluid filling that area. So if you have a swollen ankle, there's extra fluid there. That's why. Granuloma or abscess is an area of tissue holding a walled off collection of immune system cells and microbes. It's a way to kind of isolate. Yes, I understand that an abscess isn't a great feeling thing to have, but instead of that infection moving, it actually stays put in that one place. Lymphadenitis is swollen lymph nodes. I'm sure if you've gone to the doctor and you say, my throat hurts or I've coughed, the first thing they do is this business. They're actually looking to see if the lymph nodes in your throat area are inflamed. If they're swollen, that's a very good indicator that there is an infection in this area of your body. If you have pneumonia, they will usually feel under your arms because you also have lymph nodes there. There's an infection in this general area. These should swell up, and if they do, then it's a good sign that there is an infection somewhere here in the body. Leukocytosis, leukopenia, septicemia, bacteremia, and viremia. These are all terms associated with infections in your blood. So leukocytosis, cyto means cell, leuko means white. This means that if somebody were to take a blood sample and look at a complete blood cell count, they will notice that there is an increase in white blood cell number in your blood. Something that's interesting along those lines, depending on which white blood cell it is that is increasing in number, it can actually determine what kind of infection you have, which we'll learn more about in chapter 12. Leukopenia. Leukopenia means that we are lacking white blood cells. There should be high numbers and there aren't. Or you should have specific numbers even when you're not sick, and for some reason they are just not there. The numbers aren't there. Septicemia, you've got microbes present in the blood in high numbers and they are multiplying. So remember we talked about sepsis and I said it's an infection in the blood. You've got bacteria that are there, but they're not just there, they're actually replicating. Bacteremia, viremia. This kind of goes back to you've got something in your blood. It's either going to be bacteria or it's going to be viruses. The difference is, compared to septicemia, they're not multiplying now. They're there, but they're not multiplying. So if you've got viremia, there are viruses in the blood, they're not multiplying. If you've got bacteremia, there are bacteria there, they're not multiplying. In septicemia, they are. Portals of exit. How do I get out of the body if I've infected somebody? Generally, they're going to be the same as the portal of entry, but science, so not always. Pathogens generally leave the portal of exit through secretions like nasal secretions, excretions, things like human feces, discharges, or even sloughed off tissue. The more pathogens that leave through the exit, the higher the number of pathogens that use the portal, let's say a sloughed off piece of skin, the higher the number of microbes on that piece of skin, the more likely it is that it will infect the next person it comes into contact with. The transmission is more likely the higher the number that are exiting through these portals. So we've got coughing and sneezing, we've got vectors like mosquitoes, we've got skin cells or open lesions where there's infection there. We've got urine that has, a UTI. Fecal material, even the blood, HIV, in the late 60s, early 70s was when HIV really became fairly prominent enough that we realized it was there. Sadly, because it was in the gay community, nobody paid attention, even though it wasn't specific to that community. It had nothing to do with that, other than the transmission, how it was transmitted. The worst part of that was that they didn't realize it could be transferred through blood products. So if you got a transfusion, they weren't checking to see if HIV was there. If you were an IV drug user, and you shared needles, it was a quick way to get it because it was a blood product. The stages of an infection. You have the incubation period, the prodromal stage, the period of invasion, and then the convalescent period. Incubation period. It's the time from the initial contact with the microbe to the actual appearance of symptoms. The length of the period depends on how healthy the host is, what specific microbe invaded your body, what the initial dose was, and what the portal of entry was. Generally, incubation can be anywhere from 2 days to 30 days. Remember that if a microbe invades your body, it has to have a time frame to establish itself. If you get 1,000 gonorrhea microbes. Those 1,000 need to replicate in order for you to feel it. It's not enough just to have that 1,000. That's the incubation period. The prodromal stage, 1 to 2 day period where the earliest symptoms appear. Symptoms are very mild in general. I'm kind of achy, my stomach hurts, I'm tired, but it's not full blown, oh, I know what this is. It's, I don't know, I just don't feel good. That's the prodromal stage. Usually, like I said, 1 to 2 days. It's not a very long stage. Then you have the period of invasion. The microbe is multiplying and it's at high levels. So those signs and symptoms that I was talking about are now very pronounced. They're the most pronounced that they're going to be in these stages. This period can vary in length depending on the host's health as well as the specific microbe. There's a difference between getting infected with a cold and getting infected with Ebola. The convalescent period. This is when the host's immune system is responding to the infection and the symptoms start to kind of go down. You start to kind of feel better. Not at 100%, but you're getting back to 100% because your immune system is actually fighting this off. The patient's strength and health will gradually come back to normal. So convalescent means that you're healing. You are getting better. Some microbes can be transmitted only in certain stages of infection. Others can be transmitted during any point during the infection. I may not be contagious when I'm in the prodromal stage, or I may only be contagious in the prodromal stage. COVID, yeah. The scary thing with COVID is a lot of people who weren't showing symptoms yet were spreading the virus. So they were probably in the prodromal stage where they kind of generally didn't feel great, but they didn't notice that, oh, I'm totally sick. So they were spreading it. There are other diseases that until it's established, you're not going to spread it to anybody. Just depends on which one it is. Science. Terms associated with the spread of a disease. A reservoir is a natural site of the pathogen. Where does it normally reside? Is it in humans? Is it in animals? Is it in soil? Is it in water? I talked about tetanus earlier. I talked about lockjaw. That is normally found in soil. Yes, soil can get on things and you can puncture yourself with those things, but the actual pathogen is in the soil. It's not in a human. It's not in an animal. It's not a rusty nail. It literally is from soil. What is the place where that thing resides? That's the reservoir. Transmission route. How is it transmitted? Is it through direct contact or indirect contact? I have to come into contact with somebody. Directly would be like HIV. I would need to come into direct contact with someone. Indirectly may be, I didn't wash my hands after going to the bathroom. I touch a doorknob. You touch the same doorknob and then eat something and don't wash your hands before you do it. That's indirect. You still got sick, but I didn't go and put my hand in your mouth. A vehicle. This is through food, through water, through air. Sneezing and the droplets go out and you breathe them in. That's going to be a vehicle. A vector is through some type of intermediate. The most common that we kind of associate is the mosquito for things like malaria. Malaria doesn't get passed person to person, but if a person is infected, a mosquito comes and bites them, they pick it up. Things happen in the mosquito to make it mature enough that when they bite another person, it gets transferred. That's a vector. There has to be an intermediate between the two hosts. Nosocomial infections. These are infections where the hospital is the source of the disease. Nosocomial infections are hospital-acquired infections or healthcare-associated infections, HAIs, are literally acquired during a hospital stay. You don't have it, but you go to the hospital and you have it after. An average of about 5% of patients will develop nosocomial infections during a hospital stay. This leads to an increased length in the stay at the hospital, increased cost for staying at the hospital. Even it can lead to death depending on the health of the patient. The hospital is kind of the perfect storm for infections to happen. You have people that are already compromised in the hospital. They are being admitted into the hospital. You have multiple pathogens, multiple sick people, different with sick things, that are in the hospital. So the potential for having that infection around, for having multiple infections around, exists just because of the nature of a hospital. Lower defenses may allow normal biota to also invade the body. If you're already sick, your immune system's already busy. It's kind of one way that opportunistic infections can take advantage, because your immune system's already busy, let me get in now. Types of procedures used in hospitals: treatments using reusable instruments, indwelling devices like catheters or even skin grafts, and high use of antibiotics can actually increase the chances of encountering some type of antibiotic resistant strain of a disease. Again, if somebody's already in the hospital and they're being treated and it's not working, I'm going to keep trying different things. And as I try those different things, I'm selecting for things that are antibiotic resistant. It's not intentional. I don't want you to think hospitals are doing this on purpose, because that's not it. But it does happen because, like I said, it's the perfect storm. I've got sick people who are already kind of immunocompromised. I've got multiple different diseases that are floating around the hospital because I've got multiple sick people. Those people that are sick have lower immune defense. And a lot of times I'm using indwelling devices. If you've got surgeries where you catheterize somebody so that they don't wet themselves, that equipment is usually reused after it's clean. Whether it's cleaned properly is the question. What are the most common of these infections? The most common is usually a urinary tract infection, and again, it's often associated with catheterization, there we go. Surgical site infections, as well as respiratory infections, run a close second and third. So you've got urinary tract taking up 40% of this pie. You've got surgical site at 19 and respiratory at 15. Top three. What microbes are most associated with these? Gram-negative intestinal biota are recovered from samples in more than half of the patients that have this type of infection. The other half of patients are actually harboring staphylococci or streptococci, including enterococci or yeasts. It's just opportunistic that they take advantage of the fact that their immune system is kind of compromised. What can we do to control this? What can we do to control nosocomial infections? The number one method for controlling these infections: hand washing techniques, proper hand washing techniques. Other ways we can help to control nosocomial infections while working in a hospital: clean gloves are going to be used with each patient to prevent contact with the patient's blood or body fluids or transfer microbes from one patient to another. Stringent aseptic techniques when working with patients. Degerming before you inject or you catheterize a patient. You need sterile equipment and materials when performing any invasive procedure. I don't think that's a big 411 to anyone. Stringent sterilization, disinfection procedures for the rooms of the patients, for the bedding of those patients, for the breathing apparatuses of those patients. Imagine going into a hospital room, you already feel bad, and they haven't changed the sheets from the previous patient that died from pneumonia. Yeah, that wouldn't be a good thing, right? They have huge washing machines. They use bleach. There's a reason everything is normally white in a hospital, because they are doing their utmost best to make sure the room is clean, the sheets are clean, the bed is clean. All of these things have to be taken into account when you work in a hospital, because you do not want an infection going from one patient to another when you can easily avoid it by cleaning things.